Hello viewers, welcome to Biodrunks. Let's learn some aspects of biotechnology. For your reference, I'll be providing notes, PDF link in the description. This video is also available in Hindi and Telugu. For more videos and continuous learning, please like, share and subscribe. Let's start. Okay, so in the previous class, we were dealing about nucleases, where we learned how the nucleases work. So what is the work of nucleus? The nucleus is used to cut the DNA or degrade. Okay, the nucleus is used to degrade the DNA. And how it does? By breaking the phosphodiester bond. This was what dealt in the previous video. In this video, we are going to learn about DNA ligase. So what is this DNA ligase? Let's learn about DNA ligase now. Okay. So when I talk about DNA ligase, what does DNA ligase do? Okay. So DNA ligase, it joins the DNA molecules by synthesizing the phosphodiester bond between the nucleotides. Okay, or at the ends of two different molecules or a single stranded molecule. So for example, let's draw a DNA. This is one strand. This is another one. So here I have a nick present. If you could see that, okay, this is the nick present, 5' to 3', again 5' to 3'. Okay, this is 3' to 5', 5' to 3'. Okay, so as you can see how the nick is present. So what is the DNA ligase gonna do? DNA ligase is gonna attach here at the point of the nick, and then it's going to join them together. So now when you have the DNA, so this does not have any nicks present. This is also called as molecular glue. We call it as molecular glue even. Okay, so before doing this, let's learn what are the different types of DNA ligases and what is its function. Then we will go into the mechanism of DNA ligase. So DNA ligase 1, what does it do? It ligases the DNA of the lagging strand. Okay, so if you are aware of it, the lagging strand always has Okazaki fragments, that is smaller fragments. So here you have the use of DNA ligase, where the DNA ligase is gonna join these nicks. Okay, now comes DNA ligase 2 and DNA ligase 3. DNA ligase 2 and DNA ligase 3, they are helpful in DNA repair. Okay, so where are they helping? They are helping in nucleotide excision repair and to join the recombinant fragments. Now we have DNA ligase 4. What does DNA ligase 4 does? Again, it's also used in repair. Where is it? In non-homologous and joining DNA double-stranded break repair pathway. This is where your DNA ligase 4 is being used. So now we know what are the different functions of DNA ligases. Let's talk about the mechanism of action of DNA ligase. See, just now we got to know that DNA ligase mediates in the formation of two covalent bonds. Okay, where does it forms the covalent bond? It forms the covalent bond between 3' OH of one nucleotide with and 5' phosphate of the another. Okay, and it requires ATP. That is very important. It requires ATP. Now, how does it work? See, there are different steps for ligation. The first step, as you can see here, this is the first step where this DNA ligase, it binds with AMP, okay, AMP unit at its lysine side chain. So can you see it's the lysine side chain using ATP or ADP, and it forms an enzyme complex, that is enzyme AMP complex. As you can see here, this is the enzyme AMP complex. Now this enzyme AMP complex binds to the 5' phosphate group. Can you see here the phosphate group? This is the phosphate group, 5' phosphate group, okay, of the substrate DNA, okay, forming an AMP DNA, AMP 5' DNA, releasing the enzyme. Now here the enzyme is released. Okay. Now, what's the third step? Now you have this AMP 5' DNA. Now this AMP 5' DNA complex then releases the AMP. It releases the AMP and it forms a phosphodiester bond with the adjacent 3' OH. Okay, here it's forming the phosphodiester bond with the adjacent 3' OH, as you can see here. Okay, and it releases an hydrogen. Okay, our AMP, here it releases hydrogen, AMP, and this is the DNA which has been nicked. This is how your DNA ligase works. Okay, it's very simple and very easily we learned about DNA ligase. So what are its uses? So the uses of DNA ligases, it helps in DNA replication. Okay, recombination. Recombination means, see, you have the processes like gene cloning, and then you have to construct recombinant plasmids. Yeah, construct recombinant plasmids. It's where your DNA ligase comes. Recombinant plasmids and vectors. Okay. Now, DNA repair. So obviously it's used for DNA repair. Okay, as you have seen in nucleotide excision repair, DNA replication, where it is used in to form the nicks between the lagging strand. So lagging strand, it always have Okazaki fragments. Okay, because the DNA cannot do it opposite way. Okay, always the replication occurs in 5' to 3'. So this is 5' to 3', and this is called a lagging strand. Here you have the 5' to 3'. So it forms breaks. Okay, hence this breaks have to be sealed. Then that is where your DNA ligase helps. I think it's very easy, and we have covered almost all the steps related to DNA ligase here. This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to Biodrunk. Let's meet in the next video. Bye-bye.